Hello and welcome to this video. So today we will be talking about the Krebs cycle, or what we call the citric acid cycle. So what is a Krebs cycle? So the Krebs cycle is a biochemical pathway that is used to generate energy to the oxidation of acetyl-CoA. So it is also used for the synthesis of NADH and for the production of amino acids. So Krebs cycle takes place in the mitochondria of eukaryotes and the cytosol of prokaryotes. The first step involved is actually a preparatory step which begins with pyruvate. Pyruvate is derived through the glycolysis of glucose, which is a 6-carbon compound, and it is split into 2 molecules of pyruvate, which is a 3-carbon compound. The next step includes the oxidation of pyruvate into the acetyl-CoA by the enzyme pyruvate dehydrogenase complex. And in this reaction, a molecule of carbon dioxide and a molecule of NADH is generated. The acetyl-CoA is a 2-carbon compound. In the next step, the acetyl-CoA combines with oxaloacetate, which is a 4-carbon compound, to form citrate, and hence the resulting molecule is a 6-carbon compound. This reaction is catalyzed by the enzyme citrate synthase. In the next step, the citrate is isomerized into isocitrate by the enzyme aconitase. Next, the isocitrate is oxidized into the alpha-ketobutyrate by the enzyme isocitrate dehydrogenase. And in this reaction, a molecule of NAD is reduced to NADH and a molecule of carbon dioxide is generated. So hence the alpha-ketobutyrate is the 5-carbon compound, since 1 molecule of carbon is lost into carbon dioxide. Next, the alpha-ketobutyrate is converted into succinyl-CoA by the enzyme alpha-ketobutyrate dehydrogenase. And in this reaction also, a molecule of NAD is reduced to NADH and a molecule of carbon dioxide is released. Hence, the succinyl-CoA is the 4-carbon compound. So next, our next step is the succinyl-CoA, which is converted into succinate by the enzyme succinyl-CoA synthase. In this reaction, a molecule of GTP is generated. So next is succinate is converted into fumarate by the enzyme succinate dehydrogenase. And in this reaction, a molecule of QH2 is generated, which is used for the production of FADH2 fumarate. Then converted into malate by the enzyme fumarase, and for the last step, the malate is converted into oxaloacetate by the enzyme malate dehydrogenase. And in this reaction, also NAD is reduced to NADH. Now let's look at the results of the cycle. So through each cycle, the following products are generated: 3 molecules of NADH, 1 molecule of FADH2, 1 molecule of GTP, and 2 molecules of carbon dioxide. Since glucose is split into 2 pyruvate compounds for each molecule of glucose, this cycle runs twice. Hence, the product formed are 6 NADH, 2 FADH2, 2 GTP, and 4 carbon dioxide. All the NADH and FADH2 are next fed to the electron transport chain for the generation of ATP. So that's it for this video. I hope you learned something, and thank you for watching.